Hi everyone, thank you for joining today. We'll get started in about 5 minutes. Hello everyone, and thank you for joining today. Welcome to the NOAA Omics seminar series. I am your host, Nicole Miller, and I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. The seminars from this series are made available on YouTube and posted on our NOAA Omics website. There should be a link to the website in the chat now. As participants, you are muted, but feel free to type questions in the questions for staff box throughout the presentation, and at the end, I will read the questions for the presenter to answer. So with that, our presentation today is titled Cryptic Species Identification by Anita Wray. I'll go ahead and pass screen sharing permissions to Anita. Anita is a genetic data analyst working concurrently with the Genetics and Evolution Program and the Fisheries Resource Analysis and Monitoring Divisions at NOAA Northwest Fisheries Science Center. She leads research on many rockfish species, including species identification, epigenetic aging, and database management. She received her master's in science from the University of Washington, where she assessed the population structure of interspecific hybridization of 8 rockfish species in the Puget Sound, Washington. Thank you so much, Anita, for presenting today, and I'll now pass it off to you.
Amazing. Thank you for the introduction, and thanks everyone for coming today. I'm excited to talk about my work that I've been doing since I came to the Northwest Fisheries Science Center. So in general, to start off today, as mentioned in the introduction, I am primarily talking about cryptic speciation today. So to briefly introduce cryptic speciation specifically in the context of the genus Sebastes, which are rockfish, cryptic speciation is not uncommon in rockfish. There are over 100 species of rockfish within the genus Sebastes, with over 60 of them living on the US West Coast. And many of these species tend to look alike or are part of a cryptic species complex. 2 well-known cryptic species pairs are shown here on the slide. On the left is rough-eye and black-spotted, and on the right is blue deacon. You might be able to pick out some differences on the slide here when they're in beautiful lighting and they're still, but when we're talking about hundreds coming up in a trawl net, or it's your first or second time out as a recreational angler, it can be really easy to misidentify species within these species complexes. It's important to try to ameliorate this misidentification issue, as it can have a direct impact on the effective management of the 2 species. The cryptic species pair that I'll primarily be talking about today is the vermilion sunset rockfish species complex. What further complicates the vermilion sunset rockfish complex is that they're not only commonly mistaken with each other, obviously, they're also commonly mistaken with other rockfish species, mainly canary and yellow-eye. What you're seeing on the slide here on the right is a pamphlet from California Fish and Wildlife that shows just how similar the 3 species are, being vermilion, canary, and yellow-eye. This is particularly important as it is prohibited for yellow-eye rockfish to be taken or possessed in the state of California. Additionally, these species, and just orange rockfish in general, are a very popular group of fish. They happen to be a very common recreational catch, particularly in the state of California. Specifically, the vermilion sunset rockfish complex are the 3rd most commonly caught recreational species in the state. The pictures that you're seeing on the slide here are just a simple Google Images search, just to show you how common it is to pose with a photo of one of these species. So transitioning a little bit into the history of this species complex and why we know that there are 2 separate species here. It was first discovered as a cryptic species complex in 2008 from a study published by John Hyde down at the Southwest Fisheries Science Center. Within this study, they also found a significant depth segregation between these 2, what they then called types of vermilion rockfish, now formally described or informally described as sunset and vermilion rockfish. Sunset has yet to be formally described as a separate species. This study also found minimal morphological differences between the 2 species, hence the cryptic species complex. They also found evidence of introgression as they looked at both mitochondrial and nuclear loci. As a result of the minimal morphological differences that this study found, vermilion and sunset rockfish are not identified to a species-specific level in NOAA-based surveys. Vermilion and sunset rockfish are thus managed as a species complex based on the most recent stock assessment in 2021. As I've noted previously, it's important to note that this species complex is the third most commonly landed recreational species in the state of California, suggesting that vermilion and sunset rockfish play a large economic role in Southern California, and understanding their biological differences is a critical component of effective management. Expanding a bit here on the genetic basis of the first 2008 study, given that this is an omics talk, what you're seeing here is one of the figures produced in that original 2008 paper that shows the distribution of the different groups or the types that they found in this paper. They also had one of the similar species that I've mentioned previously, Sebastes pinniger, which is canary rockfish. Again, they looked at the Site B region as well as 9 microsatellite loci and had an FST between these 2 vermilion types of 0.1. What I want to highlight here in this study is that they found the type 1, so those red individuals, were found at significantly deeper depths when compared to the green individuals. So the green individuals are now vermilion rockfish, and those red individuals are now named sunset rockfish. There was a follow-up study done and published in 2022, which looked at similar species differentiation. This time they used rod sequencing and had just a slightly higher than 10,000 SNPs with an FST between the two species of 0.5, which is quite large for a marine species. And you're seeing on the PCA here, these 2 species splitting really nicely along PC1, sunset being those green individuals and vermilion being those purple individuals. What this study also found, though, is that vermilion rockfish has some pretty distinct population structure with those 3 population groups or clusters that you see along PC2. What both of these studies also found, as I've explained previously, is evidence of hybridization. You can see low-level, pretty infrequent hybridization appearing in both of the structure plots on the bottom of the slide here. Our project set out to continue this work and build a rapid identification method for this species complex. To do that, we set out to build a GT-seq panel. What we did here is we built off of the previous study published in 2022. From those 10,000 SNPs that this study used, we found 195 SNPs that had fixed differences that we wanted to look at. So fixed differences being an FST of greater than 0.99. We found 152 markers that differentiate sunset from vermilion, so explain the differentiation on PC1 here. We found 19 markers that differentiate canary from sunset and vermilion. That was mainly utilized as a QA/QC, just to make sure that we are not including individuals that were misidentified in the field. We used 19 markers that separate intra-specific vermilion groups, so those 3 population clusters you're seeing on PC2 here. And then in addition, 4 markers from a previous study. It was a follow-up study from John Hyde that was unpublished. We validated this panel with vouchered specimens to confirm that it could confidently distinguish between sunset and vermilion. We also tested some other rockfish species on this panel to confirm that it could highlight and flag misidentified samples pretty quickly. Now that we had a quick method for identifying these 2 species, we needed samples. Thankfully, the Northwest Fisheries Science Center runs 2 surveys that were of particular interest to us. On the left here is the West Coast Bottom Trawl Survey. It's an annual survey that's been going on since 1998 and occurs coastwide from Washington down to Southern California in 2 passes, one in early summer, one in late summer. It has a really wide depth range, but because it's using a trawl net, it can only sample in trawlable habitat or soft bottom habitat. On the contrast here, the Shelf Rockfish Hook and Line Survey, again an annual survey, but it's limited down to Southern California and the Southern California Bight, only in September and October, and has a much smaller depth range. But because we're using hook and line deployed by rod and reel, we're able to sample in high relief habitat, in rocky habitat, which are habitats more often occupied by rockfish. So during both of these surveys, sex, weight, and length is calculated for each fish caught, and a subsample are selected for otolith extraction for age estimation. In total, both surveys produce almost 3,000 samples annually in the vermilion sunset species complex. So now that we had a GT-seq panel that enabled rapid species identification and an annual input of almost 3,000 samples per year, I'll give you a little overview of the objectives of this project. What we wanted to look at is we wanted to examine the inter and intra-specific differences for vermilion and sunset rockfish. We wanted to look at distribution both spatially, so along the US West Coast, and then also within the water column, that being depth. We also wanted to look at key biological characteristics, as those are very important for stock assessments for these species. The ones that I'll be talking about today are age growth and length weight parameters. We analyzed these differences between 3 groups. So species specific, so sunset versus vermilion, population specific, so we looked at those 3 populations of vermilion shown in that 2022 study. And then also we wanted to look, and if we were able to identify any hybrids, we wanted to look at the biological characteristics and the distribution of those hybrids as well. I've already mentioned a lot of my methods, but in brief here, we extracted DNA from each of the fin clips of the samples using a QIAGEN kit, prepared them for GT-seq using a modified version of the Campbell protocol, sequenced on a MySeq, and then we used a package called Rubius. Rubius is an R package initially released for salmon stock identification that has been modified in previous studies for species identification in rockfish. The benefit of this package is it not only gives you a species call, but it gives you somewhat of a confidence interval that you can use to either accept or reject the species call. In overview here, we were able to get almost 31,000 fin clips. Of those, we were able to get almost 25,000 species ID calls. Before I jump into the data here, I just want to highlight simply how much effort it took to get all of those fin clips. It required collaboration over the course of 24 years from 10 chartered vessels, both commercial and recreational, that enabled those 2 Northwest Fisheries Science Center surveys to happen. We also utilized collaborations from ODFW, WDFW, and the Southwest Fisheries Science Center to kind of fill in some of the distributional gaps we have in our study. What you're seeing on the left here is the spatial distribution of the 2 species. Vermilion on the left, which you'll see will always be in purple for the rest of the talk, and then sunset will be in orange for the rest of the talk. The distribution of vermilion rockfish for our study extends from inside Puget Sound in the state of Washington all the way down to Ensenada, Mexico. The true distribution of vermilion rockfish does extend as far north as Prince William Sound in Alaska, but our samples just come from the range of where our surveys and other data sources operate. On the other hand here, sunset rockfish on the right are spatially limited to south of Point Arena. When we add in the distribution along the water column here, we see, similar to previous studies, that sunset rockfish live at significantly deeper depths compared to vermilion rockfish. On average, sunset adults live at 142 meters, where vermilion adults live at 102 meters. When we combine these 2 factors together and focus in on Southern California, which is where the majority of our samples were collected, we get a plot that looks like this, with points sized according to how many samples we caught in that area, and then colored based on the depth of capture. And what you're seeing here is for sunset rockfish on the left, you're seeing these points are not only more orange and yellow, suggesting they were caught at deeper depths, but also tend to be more offshore when compared to the shallower and more inshore vermilion. When we look at the relative proportion of each species along the water column here, we are seeing relative proportion on the y-axis here, bin depth in bins of 10 on the x, and we're seeing that there's a mix of the 2 species in depths between 41 and 130 meters. But once you get deeper into the water column here, we're seeing that sunset rockfish make up 98% of the catch below 130 meters. Moving to the biological characteristics here, we're also seeing some pretty significant differences in life history. One example here is with the standard von Bertalanffy growth curve, with length on the y-axis and age on the x-axis. We can see that sunset rockfish, shown in yellow and orange, grow both faster and larger overall. We also see significant sexual dimorphism for both species, although the pattern of sexual dimorphism for vermilion rockfish is much smaller. In Oregon, there's a lot more sexual dimorphism among vermilion rockfish, so this seems to be an effect of a large sample size coming from California, which might have a smaller sexual dimorphic pattern. Despite all of the differences that I've shown, we also found some really cool similarities between the two species. Here you're seeing plots of bin depth on the x-axis, weight on the top, and length on the bottom on the y-axis. We see that both species here experience ontogenetic migration, where the individuals migrate to deeper water as they age or get bigger. We also see that juveniles for both species live in very similar depths. We also found similarities in length distribution along the geographic range of our study. Specifically, we found that for both species, adult individuals were significantly smaller south of Point Conception or within the Southern California Bight when compared to individuals north or outside of the Southern California Bight. Size truncation may be due to a variety of factors, including increasing sea surface temperatures, decreasing values of chlorophyll A, but then also more importantly, may be due to overfishing. It's important to note that the Southern California Bight does see the highest fishing pressure for both of these species. So in conclusion, for the species-specific differences that we were able to find, we found that sunset rockfish live significantly deeper, live further offshore, have a faster growth rate, and reach a larger maximum size when compared to vermilion rockfish. Within the vermilion populations here, these three populations have arbitrarily been called VA, VB, and VC based on the 2022 paper. And you're seeing the differences in distribution here, with the VA population being kind of the northern population, ranging from the state of Washington down to Central California. The VC population, on the other hand, is limited down to the Oregon-California border, all the way down to Ensenada, Mexico. Interestingly, that VB population we saw distributed throughout the entire range of our sampling distribution. The distribution along the water column was also different. Specifically, VA, that northern population, we found at significantly shallower depths when compared to the other two populations in both subadults on the left and adults on the right, as shown as defined by length. Similar patterns also emerged in the life history characteristics of each population. Specifically, we found that that northern VA population is both significantly larger in weight and length when compared to the other two populations, which were not significantly different from each other. This is not an uncommon pattern in rockfish, where we find larger specimens typically at the northern range of the species distribution. So in general here for the three populations, we found that the VA population is the furthest north, is the shallowest and also the largest. The VC population is the furthest south and the deepest. And then VB population was intermediate for both biological and spatial characteristics. Moving on to the last group that we examined, the hybrids. We ended up finding a really small number of hybrids. We found in total 66 hybrids, which encompass 0.27% of the total samples we were able to have. The vast majority of those were F1 hybrids, 51 of them, with a handful of back crosses, including more back crosses with sunset than vermilion. This is somewhat similar to the findings in the 2022 paper, which found 0.7% of their individuals identified as hybrids. Despite the low number of hybrids we were able to identify, the biological results, at least in my opinion, are super interesting still. So you're seeing here that we did find some pretty significant overlap in the distribution for hybrids. That's not super surprising. Hybrids are more likely to occur where the two parental species are in the same area. So we're seeing most of our hybrid individuals in the middle plot here, distributed within the Southern California bight. When we add in the depth distribution, we're seeing a similar pattern with this beautiful kind of stepping stone overlap, where the two parental populations are significantly different in depth. The hybrid for the adult hybrids are kind of beautifully in between the two parental populations. We see that this trend is non-significant in the subadults. That's not surprising, as we expect the juveniles for both species to live in similar areas, so we expect the hybrids to do the same. We again see similar patterns with life history characteristics. On the left here, you're seeing a table of both fork length and weight for all three groups, and you're seeing that the hybrid individuals are statistically in between both of the parental species that are statistically different from each other in both fork length and weight. So in conclusion here, we found that the hybrids were intermediate for both depth, length, weight, and distribution, but were really, really rare, with only 66 found in almost 25,000 samples. So bringing this back a little bit into the management implications of results and results like this. On the left here, you're seeing a map of the management regions within Southern California. The red blocks here are federal marine reserves. Blue and purple are state marine reserves. And then orange are groundfish exclusion areas. So those groundfish exclusion areas are areas where commercial and recreational ground fishing are prohibited. On the right of the slide here, you're seeing again the distribution of vermilion on the top, sunset on the bottom, rockfish within the same area. And what I want to highlight for you is those groundfish exclusion areas, or those orange areas, are primarily habitats where sunset rockfish live and vermilion rockfish do not, particularly large adult sunset rockfish. Historical management going back to 2001 even more disproportionately protected deeper waters, or protected waters that primarily sunset rockfish live in. So the question remains, are current management measures equally protecting both species? And if they're not, do we need to adjust those management measures in order to better protect them, given that we manage them as a complex? So in conclusion, for this part of the project, we found that the vermilion sunset fishery is a really complex fishery to manage. Despite being one of the most commonly caught species, it is prone to misidentification and is part of a species complex. Additionally, previous management measures have been largely depth-based, which can potentially lead to differences in harvest rates. By utilizing genetic techniques and a rapid GT-seq panel, and the 2 annual Northwest Fisheries Science Center surveys, we have built a panel for the rapid identification of species, which have been used to distinguish biological differences. Results from this work has shown just how powerful it can be to get species-specific information for a cryptic species complex. This kind of work has allowed us to expand this type of work into other cryptic species of rockfish, including the rough-eye black spotted rockfish, which you see again on the left of this slide here. This cryptic species complex is of special interest as they are up for stock assessment this year in 2025. We wanted to apply a similar analysis method to this species complex to best inform the stock assessment this year. With that, we needed to find a panel that could distinguish between both species. We decided to first try a panel that was previously published by Diana Bacher when she was down at the Southwest Center, now up at the Alaska Fisheries Science Center. This panel used 95 microhaplotypes to identify multiple different rockfish species from each other. We tested this panel to see if it could identify between this cryptic species complex on both vouchered specimens as well as unknown specimens. And hopefully you can see on the PCA here, it was able to successfully distinguish between the two species, both visually in the PCA and using Rubius, which is the result of the colors on the plot here. We are currently working on getting more biological information, but we do have some distributional information that I'll share with you all today. So these two species, they do extend all the way up into Alaska, as you're seeing on the right plot here. Black-spotted rockfish, it's shown in blue here, range all the way from Southern California, which we caught a black-spotted rockfish just a handful of years ago on the hook and line survey. So extends all the way from Southern California up through Alaska into the Aleutian Islands. Rough-eye, on the other hand, which is shown here in green, is southernly limited to kind of the Oregon-California border all the way up into the Gulf of Alaska as well, suggesting that both of these two species do have a pretty significant spatial overlap. It's pretty wide distribution and pretty wide spatial overlap. But when we look at the depth distribution here, we see that black-spotted rockfish live at significantly deeper depths when compared to rough-eye rockfish. And then that one kind of outlier that you're seeing for the black-spotted rockfish, that is that Southern California sample. Again, we don't really know what it was doing down there, but cool nonetheless. When we look at relative proportion along the water column here, we're seeing kind of a flipped pattern where rough-eye rockfish kind of dominate the shallower zones, anywhere between 120 and 320 meters. And then there's a pretty significant overlap in the 320 to 420 meter depth range for both of these species. Before wrapping up today, I just want to highlight the other projects that me and my team are working on, specifically in the context of the Vermilion Sunset Complex. First, we want to expand our species-specific analyses to include species-specific maturity estimates. All of this information that I've talked about today, as well as the species-specific maturity estimates, can be used to explore species-specific stock assessments when this complex is up for reassessment in the near future. We also plan to explore predictive modeling, or be able to say with a confidence interval what species we caught based on the depth and location of capture for cryptic species complexes. We also hope to find funding to explore species-specific population estimates using CKMR. Finally, this fall, we have been working on a project to estimate age using DNA methylation, also known as epigenetic aging. In line with the rapid increase in genomic sequencing and data, epigenetic aging is becoming a rapidly cheaper and more efficient method for comparison from the traditional methods of aging, which is a lethal method, as shown on the previous slide. For example, the right image here is from a paper which used pre-existing markers in the genome to create an epigenetic clock for two species of river cod. By using we hope to use pre-existing markers like shown in this study, which can drastically reduce both the time and cost for this type of work. We are still working on the results and the methodology, and I would love to chat further with anyone on this call about the process. And I'm really hoping to have results in the next few months to share. With that, I'd like to thank and acknowledge this work was done thanks to a NOAA Saltonstall-Kennedy grant. I'd like to thank and acknowledge the survey team, volunteers, captains, and crew from the 2 Northwest Fisheries Science Center surveys. A lot of effort goes into getting those surveys to operate every year, year after year. Greatly appreciate their work, as well as the work from the WDFW and ODFW port samplers. A lot of the sequencing was done with collaborating with GT-seq, the company, so we thank them for that. And then finally, punching almost 31,000 fin clips took almost 350 hours, many of those hours being in COVID. And I want to thank and acknowledge how much effort it took to punch all of these individuals and all of the people that helped me out with that. And with that, I will accept any questions.
Thank you, Anita, for the great presentation today. Very fascinating work about this cryptic species, and I know I've learned a lot already. We'll now open it up to our participants for questions. You can type your questions in the questions for staff box on the GoToWebinar dashboard. I'll kick us off. My first question for Anita is a little bit more about the hybrids. We saw a lot of geographic overlap of these particular species, and I'm wondering why might you propose there was such a small percentage of samples?
That's a great question, and something we're definitely kind of looking more into, especially because the original study did find a higher prevalence of hybrids, but they were also looking at mitochondrial and nuclear loci, whereas our panel only looks at nuclear loci. And it kind of also goes into the question of whether our panel can distinguish between low levels of hybrids, given that we're only looking at 195 regions of the genome. Nonetheless, we do think that these two species, because they are, sunset adult rockfish are living in those offshore banks, and vermilion adult rockfish are not, that the overlap between these two species doesn't really occur during the when they're adults and when they're mature. Additionally, many previous studies have shown that rockfish in general have pretty complex mating systems, and there have been recordings of calls made from rockfish. I don't know too much about it, but it is really interesting to look into. So that might also play a role in their choice to not hybridize.
Thank you. Thanks for that, Anita. We have our first question by Kai, who asks, are there management differences among these species? That the ability to distinguish affects, or are you trying to establish the ability to distinguish them? So I think Kai might be asking here, what are the particular management differences for these species, and how might that affect our ability to manage them?
Yeah, so these two species are managed as a complex. So they're essentially managed as one species. And I think because they have such a large recreational impact, and because the morphological differences between these two species is really, really, really small, and we can't ask every recreational angler to measure the width between the eyes, which is kind of one of the distinguishing characteristics between these two species. So it will be a really important conversation when this species complex is up for assessment again, whether it is worth splitting these two species and assessing them separately, and therefore they would get separate management measures suggested in the stock assessment, or whether they'll continue to get managed as a species complex. That's going to be a really interesting conversation that we hope to have with the stock assessment team when this species is up for reassessment in the next handful of years.
Thanks Anita and Kai for the question. No more questions yet, but I'll go ahead and start us off with one more. All right, so you had quite a few areas of omics techniques that the team would like to move towards into further research. And I was wondering if you had any plans or perspectives on leveraging environmental DNA into some of these survey techniques, into future survey techniques for this particular project.
Yeah, that is something that we haven't looked into quite yet, but we hope to. I know that the surveys this year were kind of expanding the eDNA capabilities from the Northwest Fisheries Science Center surveys. I believe it's an integrated survey. And I'm really excited to kind of see where that survey takes us. And I know there is work being done right now on eDNA and rockfish species ID, specifically learning kind of where different species of rockfish occur using eDNA. Our project hasn't necessarily gotten into that yet, but it is a really, really interesting avenue that I hope to explore in later years with the kind of introduction of this new survey that I mentioned.
Thank you. Thanks, Anita. Alrighty, and it looks like we have one final question for today or comment from Zach, who says, have you leveraged the California Collaborative Fisheries Research Program for obtaining more fin clips for species? Zach also notes that as far as he's aware, there's no systematic collection of fin clips, but they routinely sample inside and outside MPAs and often pick up these species. Could be a nice follow-up activity to enable testing of MPA connectivity and effectiveness. So I'll go back to the original question that Zach had posed, which is, have you leveraged the California Collaborative Fisheries Research Program for obtaining more fin clips?
We have not yet, but I would love to follow up and get more fin clips. We do within our hook and line survey, it's not outlined in kind of the maps that I've shown here today, but we within the hook and line survey, we do sample within and outside one of the MPAs down in the Southern California Bight, but we would love to expand that work and kind of continue that work. Yeah, so I'd love to chat with Zach about that a little bit more. We also, sorry to mention one more thing, we also are working on a citizen science project in the next upcoming months to actually sample on boats and sample from recreational anglers to identify which species recreational anglers are more likely catching, just to kind of confirm what we assume they're catching more likely. So we hopefully are also getting fin clips from recreational anglers with that citizen science project. We hope to ramp that up in the spring and summer this year.
Awesome, thanks, Anita. We did have one more question that came in from Paris, who asks, how long did it take to build the GT-seq panel for these rockfish? Could you see the GT-seq being a reliable method for conservation work in the future?
Yeah, that's a great question. So the GT-seq panel was developed by a previous collaborator of ours, Gary Longo, who was on the, I mentioned on the first slide here. You'll have to ask him exactly how long it takes. I think, unfortunately, that's a question that's incredibly project dependent. What sometimes is easy about developing a GT-seq panel. So I'm building somewhat similar to a GT-seq panel with this epigenetic aging project that I mentioned. The easier or theoretically easier part is finding the SNPs that you want to look at. The more difficult part can be getting all of those SNPs to play well together in one tube or one well. Sometimes it works on the first go. Sometimes it takes months and months and months to get it to work. So it's really project dependent. I do see, but then once the panel is optimized, the amount of time and the cost it takes to kind of process all of these 3,000 samples every year is quite low compared to other methods. And it gives us a pretty high confidence in the species ID. So we can turn it around within, I think, 2 to 3 months on average, which is pretty great in terms of conservation and being able to get really rapid answers when things change in the environment with population sizes, distribution, etc. So I do see GT-seq being a really powerful tool in conservation in the future.
Thanks, Anita, and thank you for the fantastic presentation this afternoon. Lots of very interesting work regarding these cryptic species. And I appreciate your time in joining the NOAA Omics seminar series. With that, thank you very much to all who joined us today. Our next seminar series will be on Wednesday, March 19th by Allison Watts, who will explore eDNA monitoring in the National Estuarine Research Reserves and lessons on standardization, communication, and implementation. With that, thank you so much, Anita, for joining us today, and thank you all who joined us online and for the questions. Have a great rest of your day, everyone.